Thank you to the organizers for inviting me to this important meeting, this workshop on microbiome and HIV. Appreciate having the opportunity to present some of our research. Untreated sexually transmitted infections and bacterial vaginosis are the leading cause of HIV infection. 36% of women with symptomatic sexually transmitted infections or bacterial vaginosis access treatment in regions like South Africa, which practice syndromic management. However, 80% of women with STIs or BV are asymptomatic. They don't know they've got infected. They're not presenting in a way that would allow them to access the syndromic management treatment, and therefore not treated. So bacterial vaginosis, I probably don't have to go into too much detail for this particular audience, but it's a polymicrobial dysbiosis. It's not a sexually transmitted infection, although it's thought to be sexually associated. It's defined by the absence of health-promoting lactobacilli species, vaginal pH, which is normally quite acidic, raises to greater than 4.5, and it's also defined by the presence of a wide array of anaerobic bacteria, including Gardnerella vaginalis, Prevotella bivia, Megasphaeria, and many others. So currently, bacterial vaginosis is treated with antibiotics, although it's widely accepted that antibiotics are an imperfect way to treat bacterial vaginosis, and this is evidenced quite clearly by high recurrence rates. So we've recently published 2 studies. This is Anna Ursula Happel, who did her PhD with me at the University of Cape Town, in which we explored the potential of vaginal lactobacillus isolates from South African women to enhance treatment of bacterial vaginosis. And then a follow-on study from that, and also led by Anna Happel, was testing the regulatory framework within South Africa in a single-blinded randomized pilot trial using over-the-counter commercial probiotics available in South Africa. And we compared this commercially available probiotic formulation for vaginal health to standard therapy with metronidazole, Metrogel. So just, I mean, this was essentially, this very small pilot trial was just explore how probiotic formulations would be handled by our South African health regulators, SAHPRA, and we enrolled women and randomized them to either get standard of care with metronidazole on diagnosis of bacterial vaginosis by Nugent score, and or metronidazole followed by the over-the-counter probiotic, which contained Reuteri and Rhamnosus and some bifidobacterium, essentially what the same strains that would be included in the gut formulation, but it was a topically applied as well as oral combination. And what you can see here, and this is sort of not compelling data to show you that probiotics can be used to treat women for bacterial vaginosis. Really, the spread of Nugent scores in comparison to standard of care with metronidazole was not compellingly different, and most women basically went back to having Nugent scores less than 7. And the same with vaginal pH. So there was like this incremental decrease in vaginal pH by 1 month, but no significant differences. And we were also interested in how much genital inflammation was associated with this episode of bacterial vaginosis. And essentially what we found was that there was no significant difference. So, you know, this isn't a great result for a commercial probiotic formulation, but I think the important thing was, and this sort of argues to the point, that there really hasn't been a lot of development around vaginal lactobacilli strains being included in a probiotic formulation, and the probiotic we used in this trial included Acidophilus, Rhamnosus, Bifidum, and Longum. So that, you know, these aren't sort of our top hits for what we would include in a vaginal probiotic. So what Anna is shown here is compared to standard of care, there did seem to be an increase in some of the Acidophilus strains, but nothing much going on. And in particular, this group here of women were the ones given the topical probiotic and the oral probiotic. So, you know, these do seem to be present vaginally, particularly Acidophilus, but we didn't really see any compelling evidence of colonization with these organisms that were present in the probiotics that we used. When we looked at whether the probiotic was able to displace BV-associated bacteria and enhance the colonization with commensal lactobacilli that we would consider to confer healthy properties, again, you see no difference. So this isn't like an overwhelming evidence of success in this particular case. So taking on board the results from the study, we mined our biorepository at UCT from previously completed clinical trials, looking for novel African vaginal lactobacillus strains, and together with a team at UCT and headed by Anna, 57 lactobacillus strains have been isolated from 26 women presented in this paper. 10 of those crispatus, 9 lactobacilli gasseri, 18 Jensenii vaginalis and mucosae. And what we went on to do is compare the performance of these novel isolates, vaginal isolates, to ATCC reference strains for each of these species, as well as 10 lactobacillus strains, which were isolated from commercially available probiotics. And we did a whole battery of in vitro laboratory analyses, looking at the ability of isolates from women to lower vaginal pH. And this is a time course experiment comparing 6 hours, 12 hours through to 48 hours. And what we found was, compared to the probiotic that were the probiotic isolates, crispatus really showed good ability to reduce vaginal pH. We were also quite interested in the ability, lactobacilli metabolites, which are thought to lower pH, D and L lactate. And in this sort of series of experiments here, you can see that the panel of lactobacilli strains showed variable ability to produce D or L lactate. Obviously, we would be wanting to select isolates that produced both. And in particular, some of them produced much more of these products than any of the commercially available probiotic strains are available. So in this figure, you'll see that we screened the various lactobacilli strains we had on the x-axis, with the red ones being the isolates from the probiotic commercial products, the pink being crispatus, and so on. So the isolates we screened is on the x-axis, and we screened the ability to inhibit BV-associated organisms, including Prevotella bivia, which is commonly associated with bacterial vaginosis, as well as Gardnerella vaginalis. And we screened the inhibitory ability of our lactobacilli isolates against the ATCC strain of P. vivia, an isolate of P. vivia from a semen sample, and then primary vaginal isolates of P. vivia on the y-axis. And what you'll see in the example of Prevotella bivia is that there generally is good conservation of the lactobacilli's ability to inhibit the growth of P. bivia to quite a substantial extent. And this seemed to be the same in all of the primary isolates of P. bivia we screened activity against, as well as ATCC. What was more interesting for Gardnerella vaginalis, when we screened inhibitory ability of our lactobacilli against ATCC strains of G. vaginalis, in addition to 5 vaginal Gardnerella vaginalis isolates, was a really diverse, a wide difference in the ability of lactobacilli to inhibit these isolates, both at the individual lactobacilli level as well as the Gardnerella strain that was being inhibited. And essentially, of the vaginal isolates we looked at, the crispatises did seem to perform slightly better than some of the other primary strains. And in this instance, the commercial probiotics didn't do such a bad job at inhibiting lactobacilli isolates. So I'm going to end off the talk just sort of touching on a new developments within the research group. So, you know, this was the work we did with Anna was obviously a exciting pilot. It showed feasibility, that there is a justification for doing broader screening of lactobacilli isolates. And this led us to propose establishment of a platform for collaborative vaginal microbiome studies in South Africa with African partner countries that have the capacity to do regionally relevant vaginal microbiome studies and contribute vaginal lactobacilli derived from women in Africa, as well as testing these products in women with bacterial vaginosis on the African continent. With this in mind, we reached out to various centers of excellence around South Africa, Central, East and West Africa. And we've hosted a number of joint research meetings with the consortium with the eventual vision to do parallel clinical trials or observational trials to define stability of lactobacilli strains in the vagina over menstrual cycles to allow more appropriate selection of donors from who vaginal lactobacilli can be selected. As I mentioned, we started doing this quite opportunistically, and we looked at our biorepository from cohorts that we had previously enrolled. So we had samples stored in the freezer, and this included a study I did with Heather Jaspin, U-Choose in Cape Town, as well as Caprisa 082 and 084. And this work is headed by Sinai Ngapu and Leila Mansour at Caprisa in Durban. And from this initial work, which I'll show you shortly, we're launching into phase 2, which is to pilot the parallel cohort studies between Cape Town, together with the Desmond Tutu HIV Foundation, and Kemri in Kisumu. So this is a little bit of insight into the U-Choose cohort, which was a study funded through an NIH R01 between Heather Jaspin and myself, Heather being based at Seattle Children's Research Institute. And basically the clinical trial and all the analysis have been published in Christina's paper. Christina did a PhD on this work with Heather Jaspin, and Yalu Konstantinos did the immunology work with myself, but co-supervised by Heather Jaspin. So I just want to acknowledge financial contribution for U-Choose. So from those stored samples, 130 women in total were enrolled over 3 study visits over a period of 8 months, and we had vaginal swabs stored, and Heather Jaspin's lab did the microbiome sequencing. So we had 16S. They've also done metagenomes, and Annika Chicken is being supervised by Brian Cullen in my lab. So what we were focusing on from the U-Choose cohort were women with persistently L. crispatus dominated microbiota over 3 visits, with a rationale that they would harbor lactobacilli strains with desirable stability profiles. So from that huge cohort, these 3 individuals were down selected, and we have now gone on to isolate an additional 36 L. crispatus strains from these 3 women at the time point when there was just lots of crispatus, and they sort of more generally demonstrated stability. So we are essentially moving forward with these strains, contributing them to the Vaginal Microbiome Research Consortium, which is funded by the Gates Foundation. And we're very excited to have partnered with this excellent team. And we are hoping to push the agenda by contributing African lactobacillus strains into a combination live biotherapeutic for treating bacterial vaginosis that will work equally well on our continent in Africa, as well as globally in other regions. And in closing, I just wanted to acknowledge Heather Jaspin and her team at Seattle Children's, my collaborator Remy Fressot in France, who provided all the Gardnerella and Prevotella strains we used in Anna's paper, and was also co-supervisor to Anna, and then of course to our funders. Thank you very much.